This week in lab, we're going to be working on transport of materials into cells and out of cells. And actually, most of what we will be doing in lab this week is work that illuminates the concepts that are most challenging from lecture. Before I go any farther, I want to make sure that everyone knows what a solution is. We're dealing with lots of solutions this week in lab. And what's a solution? A solution always has got two parts. It has one part that is called the solvent. The solvent is usually a liquid, and in biological systems, it's usually water. A solvent doesn't have to be water, it doesn't even have to be a liquid. But in biological systems, we're usually talking about water. And then there is the solute. And a solute, or it could be more than one, is what dissolves in the solvent. So if you've got a bottle of water and you pour in one of those packages of water flavoring, then the bottle of water was the solvent. The package of flavoring was the combination of solutes. And together, what you drink is the solution. So I want to make sure that I got that. Now, why are we talking about these concepts that are essentially chemistry or physics concepts? Why are we talking about that? And it's because we want to understand how things go into the cell and how the cell pushes things out. Now, we're not going to talk about all the ways that this happens, but we are going to talk about diffusion and osmosis. Let me remind you that when we were learning about the cell, we learned that this thing, the cell membrane, is based on two layers of phospholipid called the phospholipid bilayer, right? And what you should have learned, hopefully, from either my lecture or Professor Khalid's, is that it is selectively permeable, selective permeability. Now, selective permeability, what does that mean? It just means that some things are allowed to go through the membrane and some things are not. That's all it means, all right? What is allowed to go through and what is not allowed to go through? Well, the things that are allowed to go right on through the cell membrane are small non-polar molecules. Remember, polar covalent bonds are bonds where there's this uneven sharing of electrons. So non-polar covalent bonds are when the electrons are being shared even Steven. And they are being shared very evenly between the two atoms of oxygen in an oxygen molecule or in carbon dioxide. And there are other small non-polar molecules like, well, heck, like testosterone, that's not actually that small, but still is allowed to go right across the phospholipid bilayer. Why? Because this area right here in the middle, this is kind of the barrier, and that's all fatty acids, that's all lipid. So things that dissolve well in fat will actually be able to go through the phospholipid bilayer. But lots of other things can't. You know, hydrogen ions are super tiny, right? They're just a proton. They can't go through. Anything that's got a charge, that is an ion, not allowed to go through the cell membrane, even if it's tiny. And then there are things like glucose dissolves really well in water. If it dissolves really well in water, probably can't go through the phospholipid bilayer. Now, we're going to be focusing on diffusion and osmosis, so that will mostly be looking at these molecules and molecules like them. So movement through membranes or across membranes. The cell has got different options that it can pick if it wants to bring things in or send things out. And in this lab, we focus on what are called passive mechanisms. Passive mechanisms, the cell itself doesn't have to pay to make it happen. The cell doesn't have to use its own energy molecules like ATP in order to make this happen. The cell just needs to let it happen. So I think of the passive mechanisms as sort of like using gravity to allow water to run downhill. right? If we want to get water from a pond that's up the hill and we want it to run into our house, we don't have to drive up and put it in a bucket and drive it down the hill. All we need is a pipe that goes from the pond to our house, and the water would run down the hill. That would be like a passive mechanism. Now, in the case of the movement of these molecules and atoms, we're not talking about gravity. We are talking about some of the natural things that atoms and molecules do. I would like you to please remember what the passive mechanisms are. They are filtration. Filtration is the mechanism where water pressure forced against one side of a membrane will trap big things on that side of the membrane, but let the water and the small things go through. Yeah, exactly. Filtration is like straining your pasta, okay? But that's passive because you're using water pressure to force it through. The ones we'll be focusing on in this week's lab are diffusion and osmosis. And in cells, diffusion comes in two types. It comes in simple diffusion, things that are allowed to go right across the phospholipid bilayer, and facilitated diffusion. In facilitated diffusion, we use the physics principle that powers diffusion, but in the case of facilitated diffusion, it would move something like a glucose. That's not allowed to go through straight through the membrane. Glucose is not allowed to go straight through the membrane, even if there is a force trying to push it through. So in facilitated diffusion, the cell facilitates the movement by placing a little protein tunnel that glucose can go through. That's facilitated diffusion. And then there is osmosis. And when you mention osmosis to biology majors, more than half of them will go, oh, osmosis. I hope that we'll be able to make it easy for you to understand. Okay, diffusion. So simple diffusion. I would like to make sure that you know the definitions of diffusion and osmosis, okay? They're slightly different. We'll get to osmosis. Simple diffusion is the movement of substances. Why do I say substances? Well, books say substances so that we don't have to keep saying atoms, ions, or molecules, right? So substances. From regions of higher concentration of those substances to regions of lower concentration of those substances, okay? So here we have got a beaker of water. Beaker is the word for these glass vessels that kind of look like a drinking cup, but they usually allow you to pour. So this beaker has been divided by a permeable membrane. The definition of permeable versus selectively permeable or semi-permeable. In a textbook, and it's usually a textbook or an exam, when they say it's a permeable membrane, they mean that whatever atoms or molecules we're looking at here, those atoms or molecules are allowed to cross the membrane. Permeable means that those things in our picture or in the exam question are allowed to cross. If it's selectively permeable, it generally means that one of these things, the blue, the red, who cares, one of those things is allowed to cross, but the other is not, okay? So that's the difference between a permeable membrane and a semi-permeable membrane or selectively permeable membrane, okay? So in this case, we've got this beaker, it's got this membrane, and what the scientist has done is poured pure water, they tell us this is a water molecule, pure water into the right side of the beaker, and water that's got something dissolved in it, let's just say glucose, okay, dissolved in it, right? Now, over time, the molecules of glucose that are crowded over here will move over to the right side where there are not glucose molecules. Simultaneously, the water molecules on the right side, they look around and they go, I feel kind of crowded over here. They will move over to the left side. So slowly over time, the two sides will have the same concentration of sugar and water. That is simple diffusion. And simple diffusion is something that you've got an instinct for, right? If you had a beaker of water and I said, okay, everyone, let's put a drop of food coloring in the beaker, you would roll your eyes at me because you know what's going to happen. At first, you're going to be able to see the drop as it goes to the bottom, but over time, it's going to dissolve into the rest of the water. And how does it dissolve? It dissolves by simple diffusion. In that case, there's not even a membrane in the way, still called diffusion. If I were to spray some perfume here, well, this is a video, so nothing would happen at all. But if I were to spray some perfume here, at first, someone standing by my door would not smell it, but after a minute or so, they would smell it because diffusion. I always envision that different atoms, ions, molecules, substances, that they are kind of like hyperactive little birds. And if I had a bird cage in that corner with hyperactive little bluebirds, and in that corner with hyperactive little redbirds, and they're real crowded where they are, and then I open their cages, they would just go flying around the room. Now, would they evenly distribute themselves? No, they would randomly distribute themselves. And that's the way substances in nature also behave. Now, I want you to notice that there are as many atoms on the right side as there are on the left side. So it's not that the right side was less crowded or more crowded in general. It was that the left side had more solute, the right side had more water, and each of them, redbirds, bluebirds, just kind of mixed together. That is simple diffusion. Now, what is powering simple diffusion? Oh, no. Let me start with the movement of molecules. Is it here? No. Particle movement. Good. Okay. Let me show you this. Let me turn down the volume. You won't hear it anyway. All righty. Let's look here. So in this particular image, it won't let me. I am so sorry. Okay. Turn that on. All right. In this particular image, you can see that there are particles, okay? Could be oxygen, could be glucose, could be water molecules, doesn't matter, okay? And if you see over here, we're kind of looking at them as if we can see the individual molecules. You really can't. But they're kind of jiggling around. In all of nature, those things that you learned about, atoms and molecules, they're constantly jiggling. They jiggle all the time. How quickly they jiggle and how big their movements are will depend on the temperature. So right now, the temperature is cold, but now we're making the temperature warmer. And as we make the temperature warmer, you'll notice that the molecules make bigger movements and they move faster and they start bumping so hard against each other that they move apart from each other. That is what's going on with all of our atoms and molecules all the time. They're constantly jiggling. When we talk about enzymes, that's going to end up being another important concept, that temperature causes the movement of molecules and atoms. And the higher the temperature, the faster the movement. And the slower the temperature, the slower the movement. And that is why when you boil water, it'll bounce around so much it turns into steam, it turns into a gas. All right, we will pick up here at the beginning of the next lecture.